All right, guys, welcome back. So this won't be nearly as long as the last video. Not every video will be super duper long. This video will be pretty short, or this lecture will be pretty short. The next one's a little bit longer, and then the last two are relatively short. So don't worry if the first one was a half hour, 45 minutes. This one won't take very long. You've only got 15 to 25 slides to cover this, and then we've got some tissues to talk about next. So we're going to continue with part B, which is substances, and basically just the mechanisms that the cell uses to transport substances across that membrane. So in the first chunk of chapter 3, we talked about the cell itself, the organelles within the cell division, everything related to cell and the cell and properties of a cell. Now we're going to talk about a particular property called membrane permeability. And something that is permeable is able to, or a membrane that is permeable allows certain things through and other things not. So permeability of the cell membrane just basically determines which of those substances can enter or leave the cytoplasm or the intracellular fluid. Freely permeable membranes, any substance can pass through. Most of our cell membranes are selectively permeable in some way, meaning that there are some materials that can pass, but others that can't. So like certain molecules that are really, really big can't get through the membrane, but others can. Or depending on the charge of an ion, like sodium ions are positively charged, they don't get through the membrane very easily. They have to have a transport mechanism. We'll talk about some of those mechanisms today. Impermeable membranes means that nothing can pass through. So the membrane creates a barrier, and that barrier can allow certain things through and certain things not to get through. It depends on the membrane and its properties. So like some ions, so permeability of plasma membranes, like cell membranes in your body, some of them that are freely permeable allow any of these substances to get through: carbohydrates, proteins, fats, ions, and water. Selectively permeable is what most of our cell membranes, the property that most of our cell membranes have, where some ions and water can easily get through, but things like proteins and fats can't. And then an impermeable membrane would be not permeable to any of those substances. Permeability, the ability of a substance to get through the membrane, is based on size, the electrical charge, so some positive or negatively charged substances can or cannot get through, the shape of the substance, and its solubility in lipids. So remember that the membrane is a phospholipid bilayer. So some substances like steroids are lipids themselves. So they like other lipids, and they can get through a lipid membrane pretty easily because they can basically fit through that membrane undetected and don't need anything to get it through. Materials that move into and out of the cell by passive mechanisms that do not require, so any mechanism that allows a substance to pass through the cell membrane that's considered passive does not require ATP. So things like diffusion and carrier-mediated transport we'll talk about next. Active mechanisms, if it's an active transport mechanism to get a substance from the outside to the inside or inside to outside of the cell, requires ATP. And we'll talk about these two mechanisms as well. So there's an overview slide. Here is passive mechanisms, does not require ATP. And here's active mechanisms, require ATP. So this carrier-mediated transport is both passive and active. Some transport carriers or transport mechanisms require ATP and some don't. So if it's a carrier-mediated transport mechanism, it doesn't require ATP, it's considered passive. If it does require ATP, it's considered active. Diffusion is passive. Ions and molecules are constantly in motion. They're constantly bumping into each other and bouncing off in any direction. Basically, the closer molecules are together, the more likely they are to bounce off of one another and push each other away. So the process of diffusion basically evenly distributes molecules in a solution based on this random motion. If there's an uneven distribution, there's a gradient that gets formed. based on typically concentrations. So if you think about a concentration gradient, diffusion occurs down a concentration gradient from an area of high concentration to an area of low concentration. And that's dependent upon the movement of the particles. So look at this figure here. There's like a colored cube of sugar, for example. If you place that into water, right here, everything is highly concentrated. All the molecules are really closely associated with one another. And they don't like to do that. So they'll start to bounce off of one another in random motion and start to move through the solution in which they're dissolving out to where there's fewer molecules. So right here is an area of high concentration and an area of low concentration. And those molecules will bounce off of one another and kind of force each other away from one another to where there's no particles, a lower concentration, until those molecules are evenly distributed throughout the solution. That's diffusion. Think about perfume in a room. If you're in a classroom with somebody else and they spray perfume, you don't smell it from the other side of the room. You don't smell it right away, but eventually you will, because where those particles of the perfume are really highly concentrated near the person who sprayed it, they're really low in concentration where you are on the other side of the room. Those molecules will bounce off one another and force each other towards you on the other side of the room until you can actually smell it. That's diffusion. An ion or a molecule that can diffuse across the membrane, it crosses by basically crossing the lipid portion of the membrane. Remember, the membrane is made up of a phospholipid bilayer. So the outer or the external or internal portion of that layer is hydrophilic, faces the water, or faces fluid, intracellular or extracellular, and the inner part of that membrane is lipid. So one of two ways molecules can diffuse across the membrane is either diffusing right through the lipids themselves in the membrane or passing through a channel that's embedded in the membrane. And that channel basically creates a pore for certain molecules to move through. Larger molecules can't diffuse because the membrane creates a barrier. Individual solutes basically diffuse down their own concentration gradient, and they're not affected by other solutes typically. Like the plasma membrane is freely permeable to water. So water can diffuse from an area of high or low solute concentration by way of what's called osmosis. So here's an example of this figure. This would be like a lipid, and here's a phospholipid membrane bilayer. So there's one layer, two layers. Remember, these heads of the phospholipids are hydrophilic. They like water, so they are facing the extracellular and intracellular fluid, and these little tails are lipids. Those lipids stick together, and if a molecule is able to diffuse across the membrane, it has to be hydrophobic, so it can make its way through that lipid membrane, diffuse from an area of high concentration to an area of low concentration. Or a molecule can diffuse through a membrane-bound channel. So membrane-bound channels are embedded in this cell membrane and create a pore, like I mentioned on the last slide, where molecules can move into or out of the cell. That's diffusion. And diffusion of a molecule is from an area of, again, high concentration to low, down its concentration gradient. Osmosis is unique or exclusive to the movement of water across a membrane towards an area of high solute concentration. So if the membrane is impermeable to this solute, but water can move, water will move to where solute concentrations are higher to try to even out the concentration on either side of that membrane. Facilitated diffusion is an example of passive diffusion using some kind of transport protein. So materials can move across a membrane down a concentration gradient by using a carrier protein. So some carrier protein would grab a hold of this molecule and carry it, transport it into or out of the cell, down its concentration gradient. So you're still using a concentration gradient, no active mechanisms like ATP. So movement is still down a concentration gradient, but this movement is now facilitated by way of a protein that will grab a hold of that molecule and transport it. So there's a figure illustrating those three concepts. Diffusion of a molecule of a solute down a concentration gradient, like carbon dioxide is the example here. High in concentration inside the cell, low in concentration outside the cell. Carbon dioxide can easily diffuse across cell membranes out of the cell. It is lipid soluble, so it's easily able to do that. Osmosis is the movement of water, where you've got water molecules, the larger green, or excuse me, the blue, and the solutes are in green in higher concentration outside the cell. So water will leave the cell, diffuse out very easily to basically equalize the water and the solute concentration on either side of the cell membrane. And then this carrier protein is called facilitated diffusion. Glucose is an example where it's high in concentration outside the cell here, and it'll use this transport protein. This is the carrier protein, itself will move glucose into the cell down its concentration gradient. Active transport mechanisms are a little bit different in that they require the use of ATP and the energy stored in ATP to transport these molecules. Active transport, for example, carrier-mediated transport that needs ATP, an example would be like the sodium-potassium exchange pump. This pump, if there's a pump in the cell membrane, it typically requires ATP. And ATP gets split, and that the energy stored in ATP gets donated to this pump so that that pump can pump sodium and potassium across the membrane in exchange for one another. And this active transport is able to transport molecules against their concentration gradient, not down the concentration gradient like passive diffusion. Now active transport is moving ions up their concentration gradient from low to high instead of high to low. Vesicle formation is another form of active transport or active movement. There's a couple examples here, like pinocytosis is basically moving fluid and small molecules into the cell. Receptor-mediated endocytosis basically brings specific molecules into the cell once they bind to a receptor on the cell membrane. And then phagocytosis is basically engulfing solid particles into the cell. The cell membrane itself actually surrounds these particles and basically engulfs them into the cell. And then exocytosis is the opposite. So vesicle formation, these three processes bring a substance or a molecule into the cell. Exocytosis is basically moving waste and secreting something from a cell out by surrounding it in a vesicle. It's a membrane-bound vesicle, and then exocytosing that substance out of the cell. And there's those transport mechanisms. And that's what we're going to finish with. That was a pretty quick video, but we will move on to the next here shortly. I will be lecturing next about tissues. So we'll have talked about cells, all in the makeup of a cell, transport mechanisms within a cell, and next we're going to talk about what cells make up particular tissues. So I will see you soon.